Welcome to Creative Proteomics. Today, we're introducing Luminex xMAP technology, an innovative technology changing how we study cytokines, chemokines, and other important protein molecules. Cytokines, chemokines, and growth factors are critical mediators in many biological processes, including immune responses, inflammation, cancer progression, and tissue repair. Their complex interactions often determine the outcome of diseases, making them key biomarkers for understanding pathophysiology, developing therapeutic strategies, and monitoring treatment efficacy. However, traditional methods for detecting these proteins are often limited by sensitivity, throughput, and the ability to analyze multiple analytes simultaneously. Luminex Corporation, founded in 1995, has been a pioneer in multiplex biomarker detection. The company's core innovation, xMAP technology, represents a breakthrough in multiplex bead-based assays, allowing for the simultaneous detection of multiple analytes in a single sample. By 1997, xMAP technology was recognized by Clinical Chemistry magazine as a truly clinically applicable biochip, earning the attention and approval of the scientific community. Its potential for clinical applications was realized in 2001 when it became the first and only multiplex detection technology to be approved by both the US FDA and China's CFDA for clinical diagnostics. xMAP continued to make strides in the research community, with significant studies published in Nature in 2005, solidifying its importance in scientific research. Since then, xMAP has been widely adopted across diverse fields such as immunology, oncology, and infectious diseases, making it a cornerstone technology for multiplex biomarker detection. Luminex xMAP technology relies on the combination of fluorescently dyed microspheres, beads, flow cytometry, and laser-based detection. The platform uses uniquely color-coded microspheres that can be conjugated with specific capture molecules such as antibodies, antigens, oligonucleotides, or other biomolecules to detect multiple targets simultaneously. At the heart of this technology are three main components: microspheres, each uniquely dyed to create distinct bead sets; reporter molecules that provide measurable fluorescent signals; and a sophisticated detection system that identifies and quantifies analytes. Here's how it works. Step 1. Beads are coated with capture molecules like antibodies or DNA probes. Step 2. These beads are mixed with the sample, allowing target analytes to bind to the capture molecules. Step 3. A reporter molecule, labeled with a fluorescent dye, is added to bind the captured targets. Step 4. The beads are run through a Luminex analyzer, which uses lasers to identify the bead type and measure the reporter fluorescence. The detection system uses two lasers. One identifies the bead type, while the other quantifies the reporter signal. The results are then analyzed, giving precise concentrations of each analyte in the sample. It's important to understand how Luminex xMAP technology compares to other common protein detection methods. Let's take a look at some of the key differences. SIMOA, or single molecule array, is renowned for its exceptional sensitivity, capable of detecting molecules at the femtogram (FG) level. This makes it particularly valuable for neuroscience research, where low-abundance biomarkers are often critical. However, despite its impressive sensitivity, SIMOA has notable limitations. It offers low throughput and comes with high costs, making it less suitable for large-scale studies or routine assays. In contrast, Luminex xMAP technology, while slightly less sensitive, provides high-throughput capabilities, allowing for the simultaneous detection of up to 80 analytes in a single sample. This makes it an ideal choice for large-scale screening and biomarker discovery. MSD technology utilizes electrochemiluminescence to achieve high sensitivity and a wide dynamic range, making it suitable for protein quantification and cytokine detection. It also minimizes matrix effects, which can interfere with detection. However, MSD requires specialized and often expensive equipment, which can be a barrier for many labs. Luminex xMAP technology also offers a wide dynamic range and can detect multiple analytes in various sample types, but at a lower cost and with greater flexibility in terms of throughput. This makes Luminex xMAP more accessible for a broader range of research applications. Microarrays are designed for high-throughput screening and can analyze thousands of proteins simultaneously, making them a powerful tool for proteomics and drug discovery. However, the complexity of data analysis in microarray technology, coupled with its high cost, can be a challenge for many researchers. In contrast, Luminex xMAP technology strikes a balance between throughput and ease of use, offering multiplexed analysis without the extensive data processing requirements of microarrays. This allows for faster, more straightforward biomarker analysis. ELISA is one of the most widely used techniques for protein detection, known for its high specificity and simple operation. Its quantitative nature and availability of kits make it popular for both clinical diagnostics and research. However, ELISA has several limitations. It offers low resolution and sensitivity, a narrow linear range, and only allows for the detection of a single analyte per assay. Additionally, it requires a high sample volume and has high variability between tests. Luminex xMAP technology surpasses ELISA in many of these areas, offering higher throughput, a broader dynamic range, and the ability to detect multiple analytes simultaneously from a smaller sample volume. This makes xMAP an attractive option for researchers who need to maximize information from limited samples. Luminex xMAP technology combines the strengths of various platforms while minimizing their drawbacks. It is highly flexible, accommodating a wide range of sample types, and provides reliable results with a broad dynamic range, typically spanning over 3.5 logs. At Creative Proteomics, we utilize the Luminex 200 instrument system, capable of processing up to 12,800 assays per hour and reading a full 96-well plate in under 45 minutes. This magnetic bead-based platform measures up to 80 analytes from just 50 L of sample, supporting a wide range of matrices including serum, plasma, tissue lysates, and culture supernatants. With its low sample volume requirement and sensitivity down to picograms per milliliter, the Luminex 200 is ideal for accurately quantifying low-abundance cytokines and biomarkers, even when sample availability is limited. Having explored why Luminex xMAP technology is the preferred choice at Creative Proteomics, it's clear that its high-throughput capabilities, multiplexing power, and versatility make it an invaluable tool for a range of biomedical applications. Now, let's dive into how Luminex xMAP technology is being applied across various research fields. In tumor microenvironment studies, cytokines such as interleukin-6, interleukin-10, transforming growth factor beta, and vascular endothelial growth factor are key mediators of immunosuppression and angiogenesis. Luminex xMAP facilitates the simultaneous quantification of these factors, allowing researchers to dissect how tumors evade immune surveillance and establish protumorigenic niches. For immune checkpoint blockade research, profiling cytokines like interferon gamma, tumor necrosis factor alpha, and interleukin-12 provides critical insights into T-cell activation and functional polarization. For example, studies utilizing Luminex xMAP have shown how PD-1 or PD-L1 inhibitors modulate these cytokines to reinvigorate exhausted T-cells, offering mechanistic data on treatment efficacy. In combination therapy development, multiplex cytokine analysis is instrumental in evaluating synergistic effects. For instance, assessing changes in cytokine profiles such as granulocyte macrophage colony-stimulating factor, interleukin-2, and interleukin-15 in response to CAR T cells or oncolytic viruses provides evidence of enhanced immune activation and tumor clearance. Additionally, the technology has been used to uncover biomarkers of resistance, such as elevated levels of interleukin-8, which correlate with poor responses to checkpoint inhibitors. By integrating these findings, researchers can better understand resistance mechanisms and identify combination strategies to overcome them. Finally, Luminex xMAP supports longitudinal studies by enabling the monitoring of cytokine dynamics over time. For example, in murine tumor models, tracking changes in interleukin-1 beta, interleukin-1 beta, and CXC motif chemokine ligand 10 has elucidated temporal patterns of immune cell recruitment and inflammation during therapy. Infectious disease research plays a crucial role in understanding how the immune system responds to pathogens. During infection, the body releases a cascade of cytokines that coordinate the immune defense. However, an overproduction of these cytokines can lead to harmful inflammation, as seen in conditions like sepsis. Luminex xMAP technology offers an advanced solution, enabling researchers to simultaneously measure multiple cytokines, such as interleukin-1 beta, interleukin-10, and interferon gamma, in response to viral, bacterial, or fungal infections. By tracking these cytokines over time in animal models or cell cultures, scientists can gain a deeper understanding of the immune response, identifying key signaling molecules involved in pathogen clearance or immune evasion. In vaccine development, Luminex xMAP is a valuable tool for evaluating immune responses to different vaccine formulations. Profiling a broad range of cytokines allows researchers to determine whether immune responses are skewed towards a T helper 1 or T helper 2 pathway, providing critical information for optimizing vaccine candidates and enhancing their efficacy. In autoimmune diseases like rheumatoid arthritis, lupus, and multiple sclerosis, the immune system mistakenly attacks the body's tissues, with cytokines playing a central role in driving inflammation and tissue damage. In rheumatoid arthritis, elevated levels of tumor necrosis factor alpha and interleukin-6 promote joint inflammation and destruction. In multiple sclerosis, cytokines such as interleukin-17 and interleukin-23 activate pathogenic T cells that target the central nervous system. Luminex xMAP technology is widely used in autoimmune disease research to profile cytokine networks in both preclinical models and patient samples. By measuring key cytokines, researchers gain insights into dysregulated inflammatory pathways. For example, interleukin-1 beta and interleukin-18 drive inflammation in conditions like gout and systemic lupus erythematosus, while interleukin-10 regulates excessive inflammation. Luminex xMAP also helps assess the balance between pro-inflammatory and anti-inflammatory cytokines, crucial for understanding disease mechanisms. In rheumatoid arthritis, the ratio of interleukin-17 to interleukin-10 reveals the extent of immune dysregulation, with higher interleukin-17 levels contributing to tissue damage. Additionally, Luminex xMAP supports the development of targeted therapies. By profiling cytokine responses to treatments like biologics targeting tumor necrosis factor alpha or interleukin-17, researchers can evaluate their impact on immune modulation, treatment efficacy, and disease progression. This technology also aids in identifying potential biomarkers, such as high levels of interleukin-21 in lupus, which correlate with disease activity and can be used to monitor patient responses. Neuroinflammation is central to the progression of neurological diseases like Alzheimer's, Parkinson's, and multiple sclerosis. Cytokines, such as interleukin-1 beta, tumor necrosis factor alpha, and interleukin-6, play a significant role in inflammation and neurodegeneration. For example, in Alzheimer's disease, elevated interleukin-1 beta levels are associated with neuronal damage and cognitive decline. Luminex technology allows for precise monitoring of neuroinflammation by measuring cytokine levels in brain tissue or cerebrospinal fluid. In studies of stroke, for instance, Luminex has shown significant increases in tumor necrosis factor alpha and interleukin-6, which correlate with neuroinflammation and brain injury. This technology helps researchers understand how inflammation in the brain drives diseases like Parkinson's. In preclinical studies, Luminex is used to assess the impact of neuroprotective compounds on inflammation. For example, in Parkinson's disease models, compounds like ropinirole have been shown to reduce tumor necrosis factor alpha and interleukin-6, helping to protect neurons and reduce inflammation. Finally, Luminex is key to identifying potential therapeutic strategies. In clinical trials, drugs like canakinumab, which inhibits interleukin-1 beta, have demonstrated improvements in cognitive function in Alzheimer's patients by reducing inflammation. By evaluating how drug candidates alter cytokine profiles, researchers can identify promising treatments for neurodegenerative diseases. In summary, Luminex technology helps monitor neuroinflammation, understand its role in diseases, and accelerate the development of therapies to reduce inflammation and protect the brain. Chronic inflammation is a key feature of metabolic diseases such as obesity, type 2 diabetes, and cardiovascular diseases. Cytokines like interleukin-6, tumor necrosis factor alpha, and monocyte chemoattractant protein 1 play a central role in insulin resistance and systemic inflammation. Elevated levels of tumor necrosis factor alpha, for example, are linked to impaired insulin signaling, contributing to metabolic dysfunction. Luminex technology is a powerful tool for profiling circulating cytokines in preclinical models of obesity or metabolic syndrome. By using Luminex, researchers can explore how inflammation influences metabolic health, allowing for a deeper understanding of the molecular connections between inflammation and metabolic disorders. This cytokine profiling helps identify key molecular pathways that drive insulin resistance and other metabolic issues. Such insights guide the development of therapeutic strategies aimed at reducing inflammation and improving metabolic outcomes. Luminex also allows researchers to evaluate the impact of dietary interventions or drug candidates on cytokine profiles. For instance, studies have shown that certain dietary changes can lower interleukin-6 and tumor necrosis factor alpha levels, leading to improved insulin sensitivity and better metabolic health. Ultimately, Luminex technology supports the discovery of novel therapies for managing obesity, diabetes, and their associated cardiovascular risks. By monitoring changes in cytokine levels, researchers can identify promising new treatments that target inflammation to improve overall metabolic health. At Creative Proteomics, we offer customizable Luminex panels that can be tailored to your specific research needs. Our panels cover a wide range of disease models and biological systems, allowing for the detection of key biomarkers across various conditions. Whether you are studying cancer, infectious diseases, or autoimmune disorders, our Luminex xMAP-based services provide reliable, reproducible data to support your research. We can analyze a variety of species, including human, rat, mouse, non-human primate, rhesus macaque, crab-eating macaque, canine, equine, feline, porcine, rabbit, and guinea pig, ensuring that we meet the unique demands of your study. We recognize that obtaining precise and comprehensive data is essential for advancing your research. That's why we are dedicated to providing multiplex cytokine detection solutions supported by years of expertise and innovative technology. To discover how our Luminex services can help you achieve your research objectives, we invite you to explore our website or reach out to us directly. We look forward to partnering with you in your research journey.